welcome everyone. Hello. Thank you so much for coming this evening. Thank you so much for being interested in the Blean project and for being interested in the monitoring. It's a very vast topic, and I'm not entirely sure how long I'll be talking for, but hopefully we'll squeeze it into that 1 hour, including questions and answers. If you do have any questions throughout the presentation, if you could just pop them into the chat, that would be great, and then we can pick up everything up at the end of this. But yeah, I'll just jump straight in. I'm not going to talk an awful lot about the project in general. So if you are interested in what it is about, or you don't know an awful lot about it, feel free to either have a look on our website. There's a lot of information there, or there's also talks like these, but more generic about the Blean project. So keep an eye out for those over the course of the year, and come to one of those if you're interested. So this is a map of the project area. And you can see, so just for reference, this, you're not going to see that grey line here is Thornton Wood Road, which runs pretty much north to south. And then east of Thornton Wood Road, you have some lighter green areas and some darker green areas. And then you have this dark green area west of Thornton Wood Road. And those are our 3 treatment areas. Excuse me. So the lighter green areas are what contain our bison assemblage. So this is made up of bison, longhorn cattle, and iron-age pigs. The darker green area east of Thornton Wood Road is the domestic assemblage, made up of, again, exmoor ponies and iron-age pigs, but alongside longhorn cattle in place of bison. And then everything west of Thornton Wood Road is our control area. So there's no grazing animals in there whatsoever. This is an experimental approach and what is called a before-after control impact study design. And what's really exciting about this from a monitoring point of view is that not only will we be able to see what changes over time and how that changes, but we'll be able to compare that very unique behavior and ecology the bison have and the impact that has on the ecosystem with a, similarly large grazing herbivore, the longhorn cattle. And also we'll be able to compare all of those impacts to an area that doesn't have any grazing animals that benefits from a more traditional conservation management. So this is going to be what we'll look at over the course of the next 5, hopefully even 10, or maybe even more years. And ideally we'll be able to gather a lot of evidence of how these nature-based solutions work and how they help restore our ecosystems and make our woodlands more climate resilient. Right, in terms of the monitoring program, so just to slowly slide into this and again give you a bit of an overview of what we're doing in terms of monitoring. Our main objective is to evaluate how reinstating naturalistic grazing drives landscape scale ecological processes and integrity. And the monitoring program is divided into 4 big topics. The first one is habitat structure and vegetation dynamics, under which we're looking at things like plant community composition, successional vegetation types, vegetation age class and structure and heterogeneity, and natural regeneration. And under that topic, we're looking at all sorts of vegetation and habitat monitoring, like plant surveys, national vegetation classification, and also remote sensing. But we'll go into detail around all of these individual surveys in a minute. Then category 2 is biodiversity and bioabundance, where we're looking at anything around species, so species richness, diversity and abundance, trophic complexity, and flagship species. And that category contains all of our species surveys. So from our invertebrate surveys over breeding birds and butterflies, European protected species, all of that sits underneath biodiversity and bioabundance. Category 3 is natural processes, where we're looking at naturally driven stochastic events and disturbance. We're looking at how we can measure reducing human influence and impact, and how we're creating underrepresented niches. And that contains things like wild herbivore surveys, looking at existing grazing and browsing pressure from those. This includes heath fritillary surveys, which are flagship species of the Blean, and also movement and use of the Blean area by our introduced grazers. And then last but not least, ecosystem services and natural capital, where we're mainly looking at soil health, carbon sequestration, and pollination. So this contains most of our soil surveys, things like soil carbon surveys, also soil eDNA sequencing, and then things like pollination will come out of other surveys like the Standardised Invertebrate Survey. So before I go into each of those 4 categories in more detail and give you an overview of what we found, I just feel the need to point out that, and hopefully by the end of this talk, you will appreciate how big the monitoring program is. There's absolutely no way we could do this on our own. So not only do we rely on our own Kent Wildlife Trust volunteers, we rely on collaborations with ecology groups and their fantastic volunteers, and we also rely on partnerships with academic research partners like universities, who obviously have their own research interests, and that really benefits our monitoring program. So we're working with Canterbury Christchurch University, for example. They've been running an internship in the summer of this year, taking soil and dung samples, and they had a couple of student interns who then extracted the DNA from the soil, and this is now being sequenced, and we're probably going to look at things like soil fungus, soil bacteria, soil nematodes, things like that. The University of Huddersfield are doing aquatic diversity surveys, so they've come down for a few pond surveys so far. Royal Holloway are looking at pollen and also dung beetles, so they've got pitfall traps out and also taking dung samples from the bison to look at dung beetles. DICE are hoping to establish a big camera trapping project, which is probably going to span over the wider Blean complex, so including all of the Blean woods rather than just our project area, which is West Blean and Thorndon Woods. The University of Sussex have done a dissertation project for a couple of students, including audio moths in the Blean, so they're looking at bioacoustics. The University of Kent are doing dung sampling for microbiome and bison gut health. The Natural History Museum, we're biobanking our soil with them, so there's a whole heap of things that they could look at in terms of soil analysis that we currently don't really have the funding for. But instead what we've done is collected 145 soil samples as part of the baseline, and we're biobanking them with the Natural History Museum at the moment to hopefully come up with that funding in the future and be able to do a little bit more around soil analysis. And then obviously this is quite a new one, but there's a student project that might come out of a collaboration with them around how humans and dogs, so dog walkers, could replace predators in a landscape of fear scenario. So we're just looking at how bison and longhorn cattle react to dog walkers, and could they replace predators in a more wild landscape. And then super importantly, we're working with local ecology groups like Kent Reptile and Amphibian Group, who are doing all of the reptile monitoring on site, Kent Mammal Group, who are doing the dormouse monitoring on site, and then Butterfly Conservation, who are doing the heath fritillary counts. And yeah, as I've mentioned, there's a lot of volunteers involved, and I know that some of you are listening tonight, so let me just take the opportunity to say a big thank you to all of you. We literally couldn't do it without you. It would be completely impossible. So moving on to the first big topic, habitat structure and vegetation dynamics. Under that, we're doing the vegetation monitoring, which takes up the better part of my summer every year. So we have 145 vegetation monitoring points. They're each 300 square meters big. And in each of those points, we record pretty much anything you could imagine around vegetation. Things like species abundance and diversity, of course, also seedling counts, sapling counts, tree height, tree girth, deadwood volume, nectar abundance, nectar diversity, bramble cover, you name it. Then alongside that, we're doing plant community mapping, also called National Vegetation Classification Surveys, NVC. And whilst the vegetation monitoring is happening every year, the NVC happened last year as part of the baseline and is going to be repeated in 5 years from that, so 4 years from this year. And then alongside that, in terms of vegetation, we're doing remote sensing. Now, bear with me, I'm not a GIS and remote sensing expert, and I'm really not techie enough to understand a lot of this, but we're doing drone flight surveys, I get that. And whenever we fly a drone, we have two sensors. One of them is responsible for normalized difference vegetation index. And what that means is basically a greenness index. So we can figure out how green that particular area is and how that greenness changes over time. And then the other sensor is responsible for generating 3D vegetation modeling for us. So before we go into actual results, I just want to point out that projects like this don't always plan out as we'd like them to. So ideally, the bison would have arrived at the beginning of this year's survey season, which would have meant that last year was our baseline, and this year would have been the first data collection and the first records that actually show bison impacts or impacts from the other grazers. That didn't happen. The bison have arrived at the end of the summer this year. The other grazers are still not on site. So what that meant is that we now have 2 years of baseline survey. So everything that was done this year was either done before the bison arrived or outside of the area that the bison are currently in. So we have a more extensive, more thorough baseline, which is absolutely no problem. And it just means that everything exciting in terms of data is going to start as of next year. And so all of the data that I'm showing you tonight is either from last year or from this year, depending on how far we've gotten with this year's data, some of which I haven't received, some of which hasn't been analyzed yet, some of which is still ongoing in terms of this year's survey season. But I'll obviously point out whenever we look at data where that's from and what it is exactly we're looking at. But there will be, I'm sure there will be another monitoring talk next year. So if you're interested, please come along to that. That's when we actually get into the nitty-gritty of bison impact, and that's when it gets really exciting. Vegetation monitoring, that actually brings me to another techie problem. But again, I can't explain to you because I'm not techie enough. But all of our species data goes into what we call Recorder 6, which is a database that also talks to Kent and Medway Biological Records Center. So everything we record goes directly to them, and they can see what we do and we can see what they do, which is very beneficial. But unfortunately, there's been a glitch, which means that I currently can't see the species data for 2021, which means that I can't write the report for that year. And it means that I can't compare the species from last year to this year. We will work that out. And at the end of the presentation, I'm going to show you a link to our monitoring webpage. You can always have a look on there. That contains all of our data, all of our monitoring reports. So if you're particularly interested in the vegetation, they will get written and they will end up on that monitoring webpage. So feel free to have a look. I can always let you know via email when they're ready, and you will be able to read about the vegetation monitoring in more depth. But our data team has been kind enough to supply me with some maps just so we can have a look at a few things. Now, what you can see here is my 145 monitoring points in 2021 and in 2022. And the red crosses are the ones that I couldn't access. And you can see there's a lot more I couldn't access in 2021. I think there was about 17, whereas this year it was only 11. And that's mainly due to forestry work and tree felling that's gone on site, which meant that some areas were just a little bit more accessible. There were also extraction routes where the trees have been taken out that actually allowed me to get to some more monitoring points than I was able to last year. This is what the seedling count maps look like for last year and this year. So again, in each of those monitoring points, I count the seedlings, which means anything between nothing to 1.30 meters. We record the height and the species, and you can see that actually there isn't an awful lot of difference between 2021 and 2022. There are a few points that were bigger last year than this year and vice versa. And this is kind of what you would expect to see. There isn't anything in particular that I would put that down to, other than, again, the forestry work that has been happening on site, and also the forestry work that happened the year before, which probably took a bit of trees out and allowed more sunlight to come to the floor, allowed more seeds to disperse, which meant that there might be more seedlings in some areas this year than there was last year. But then if you look at the bottom here, this is what's called a box plot. And these lines in the middle are the median for that particular area. And actually, if you compare the medians between the two treatments in terms of seedlings, there really isn't an awful lot of difference. And actually, fairly similar for the saplings. Again, some of those points were slightly bigger this year. Some of the points were slightly bigger last year. This is very much what we'd expect to see. And hopefully those are the kinds of maps that are going to change considerably over the next few years. And those are the kinds of maps that will be really exciting once we see the impact the bison are going to have in all of their tree taking down abilities. And then last but not least, my colleague Lawrence has gotten this together for me, which shows you the tall herb abundance. So part of the vegetation monitoring is to have a look at all the layers in the woodland so that we have an idea of what the layering looks like, what the structure looks like. So this down here is called a day-4 scale, which goes from dominant, abundant, frequent, occasional, rare, to not there at all. And we do that for not only every species that we find in each vegetation monitoring point, but also for the layers of the woodland. So we're doing that for bare ground, short grass, medium grass, rank grass, tall herb, which is what we're looking at here, low scrub, medium scrub, established scrub, and then established trees or woodland. Again, nothing much to report here. There's some differences in terms of there was more tall herb in this area than there was over here, and vice versa up here, which could be down to all sorts of reasons, but nothing that is crazy unexpected. But again, this is going to be something that will be really interesting to look at over the next 5-10 years. And again, this is exactly what we want the bison to do. We want them to increase the structural heterogeneity, to increase the amount of layers, so that, you know, if you think about a pine plantation, all you have is that really high tree layer, and then underneath there's usually a bracken layer or bramble layer, which is all at the same height. Not very diverse, not very great habitat. So ideally, we just want more layers. We want a bit more of everything. We want more heterogeneity. Okay, moving on to National Vegetation Classification Survey. I appreciate this isn't particularly easy to see because it's all just very green, but that's because it's all quite similar when it comes to looking at it from an NVC point of view. So this was carried out by ecological contractors. And the outcome of this is that the study area supported 557 hectares of woodland, which is equivalent to 98% of the total area, which is kind of what you would expect. And the most common and widely distributed NVC woodland type was W10. That's an oak, bracken, bramble community, which accounted for 85% of the woodland present, with subcommunities W10A and W10B. And what they mean is it's basically still the same community, but they're slightly degraded because they have pine plantations on them. There was also some low-lying areas with base-rich soils, typically supporting W8, which is beech and bramble, W1, that's grey willow, marsh bedstraw, and W22, which is a blackthorn bramble scrub community. Now, for those of you who might not be very familiar with NVC surveys, when I mention those species like W10, which is the most common, oak, bracken, bramble, that does not mean that all you find in that area is oak, bracken, bramble. In fact, you might stand within a W10 plot and there is no oak, bracken, or bramble, but there's other indicator species that will tell the surveyors that this is a W10 community, and those 3 species are just used to describe W10. It doesn't actually give you that much insight. And then this is just a little drone flight video of what the GIS team have been up to. So we have 3 drone transects in each treatment, so 9 altogether, and they flew them this summer. And as I've mentioned, there's 2 sensors on those drones. One is called multispectral sensor. The other one's called a photogrammetry sensor. The multispectral one is responsible for the for the NDVI, so that's the greenness index, and the photogrammetry is responsible for the 3D vegetation modeling. None of that has been done yet, unfortunately, so I can't include that in this talk, but again, please check back. It will be done, and it will be something really nice to look at. But yeah, the baseline transects were flown in the summer, which is the main thing, before the bison arrived, and now the team will be busy over the winter to analyze and create all these models. That takes me on to category 2, biodiversity and bioependence, under which we're doing the standardized invertebrate survey. I say we, they're being carried out by Graham Lyons. For those of you who don't know him, he is an invertebrate and vegetation expert who does a lot of monitoring sorry, on a lot of wilding sites in the UK, and uses all sorts of vegetation sampling. You could think of suction sampling, sweeping, beating, log turning, flower searching. He did that very extensively last year, again, as part of the baseline, and this is something that won't be repeated every year, but we'll do that again 5 years from the baseline, so in 4 years' time. Botanical richness, diversity, and abundance is going to come out of the vegetation monitoring. Breeding bird assemblages and lesser spotted woodpecker surveys are being done by ecological consultants for us, with the breeding birds being done on an annual basis and the lesser spotted woodpeckers being done every 5 years again. UKBMS is the UK butterfly monitoring surveys, which happen over the summer, and they're being carried out by volunteers once a week. European protected species, I already mentioned CRAC, who are doing our reptile monitoring between March and October. We've got Kent Mammal Group, who are doing the DOMAS monitoring between March and November. And then we are also in the process of establishing a little audio moth project. So this is bioacoustics. These are tiny little sensors that record sound data. So we're hoping to be able to put them out over the next few weeks to look a little bit further into nocturnal birds and also bats. Bioabundance is mainly done through invertebrate abundance. So we have, these are called slam traps out in the Blean between April and September. They're being serviced once a month, and all the invertebrates that are found in there are being analyzed by a heap of fantastic volunteers who go through them in the winter, and they sort them by order. So we're not looking at diversity here, we're looking at a number for abundance. So how many flies, how many bugs, how many beetles. And then last but not least, trophic complexity, which is something that will come out of all sorts of other surveys like the invertebrates. Okay, results for the standardized invertebrate survey. So in the top left, you can see the map. Graham had 8 monitoring points per treatment area. And right underneath the map, you can see a pie chart of what he found most commonly. Started with Coleoptera, which are your beetles, closely followed by spiders, a lot of moths as well, bugs, then Diptera, which are your flies, Hymenoptera are your bees, wasps, and ants, butterflies, and then slowly going into the smaller orders. In terms of really exciting things he found, and please bear with me, I'm not an entomologist if I pronounce any scientific names correctly or funnily. In the top left corner is Pistius truncatus, which was last seen in the UK 20 years ago, but it's in the Blean, so that was a very exciting find. Top right is Vulcanaria mitrata, which was last seen in the UK in 2004, so nearly 20 years ago as well. Bottom left is Dicelucas niverostris, which is a rare anthrobit weevil, so a fungus beetle, that hasn't been seen in the UK since 1988. Graham Lyons found it in the Blean last year. And then bottom right is Lagria atripes, which is regionally extinct, but actually doesn't seem to be very rare in the Blean at all, which is fantastic. And then underneath that, and I appreciate you might not be able to see that very well, but it doesn't really matter. All I wanted to show you with that little table is that it's really nice to see how all 3 treatments, so at the top you can see C for control, B for bison, P for proxy, all 3 treatments are actually quite similar no matter what you look at. So the species number is really similar, and then if you go into the different orders, all of those numbers are really quite similar as well. If I just pick out butterflies, 10, 15, and 12, or Diptera, your flies, 39, 36, 31. So that's great because it just gives a really nice baseline to then look at how things change over the next few years. What changes first? How does it change? What changes next? What takes longer to change? What changes quickly? And so on and so forth. Next up is our birds. So you can see in orange, blue, and purple the transect lines. There's one in the control, 3 in the bison area, and then another 3 in the grazing assemblage area. And then the colored dots around those lines are your lesser spotted woodpecker points. So where the dot is really quite small, the surveyors found or heard 1 lesser spotted woodpecker. Where it's slightly bigger, it was 2 birds. And the biggest point shows you 3 birds. And so for 2021, the surveyors found 34 breeding bird species that sort of represent the range of your typical woodland birds. Most numerous were robins, bluetits, wrens, and cheffins, the kind of thing you would expect. But they also included a number of red-listed species that were recorded in smaller numbers, like nightingales, spotted flycatchers, and lesser redpolls. The survey for this year was very, very similar assemblage to 2021, with the only difference that they only recorded 31 species. They didn't hear any lesser spotted woodpeckers, any lesser redpolls, and any rooks. And in terms of lesser spotted woodpecker surveys that were done in depth last year, there was a minimum of 13 individuals recorded during the transect surveys, with a minimum of 3 further individuals found outside of these timed transect sections. So it is considered that the 16 plus birds located during the survey are likely relating to at least 8 pairs, and that the total population of the site is likely to be in the order of 10 to 16 pairs, which is really lovely. So that was a very nice baseline outcome. Moving on to butterflies. Again, you can see the transects on the map, and then in the table on the right are the results from 2021, because I don't have all of the data for this year yet. I'm still waiting for 1 transect. And you can see actually there's quite a bit of difference between the transects. So West Blean Wood West, West Blean Wood East, and Coalwood, which are these 3 transects, sort of more in the southeast, had a lot higher numbers than the other 2 over here. Now that can be down to so many reasons, including that surveyors may not have gotten out as often, that they weren't able to take a picture. You usually have to submit the photo with every record you submit. You know, so this doesn't necessarily tell us that there's less butterflies in this area than there is down here, but definitely something that we'll keep an eye on over the next few years and see how that develops. And in terms of species, so the ones with the highest counts were ringlets, meadow browns, gatekeepers, red admirals, and then really lovely to see our heath fritillaries, because they are flagship species in the Blean. And actually this year, looking at the different transects, as I've said, I'm still waiting for data for 1 transect, but they all look fairly similar. The numbers are pretty close to what they were last year, with the exception of West Blean Wood West. This one here, they had a record of 1,696 butterflies, so pretty much double, which is really nice and super lovely to see. And this year, additionally to the ones I've already mentioned, with the high numbers, they're all still in high numbers, but we also had brimstone and speckled wood in very large numbers, and we had a new species, which was the brown argus, that hadn't been seen in 2021 at all. Dormice. The map shows you sort of the rough survey locations. I didn't want to go into any more detail than that, but we have 20 nest boxes out in each treatment area, and they're being monitored monthly between March and November by the Kent Mammal Group. In 2021, we had 3 nests in the bison area and 1 nest in the control area. Now, they were all found during winter clearance at the beginning of this year, but they are very much thought to be from last year's active season. And for 2022, we've already had 7 nests in the bison area in September, 2 dormice in May, 1 in July, 2 in August, and 4 in September. Now, the survey season is still ongoing for dormice, so there's more records to come in for dormice, probably at the end of this month. And I haven't had any records yet for the control area or the grazing area. I know that until now, normal dormice had been found in those areas, but there might be a few more nests. And actually, with there being more monitoring days, there might actually be more dormice. But this is where we're at the moment. And reptiles, again, you can see in the map sort of the rough survey locations. Again, I didn't want to go into any more detail, but this is where Crag have their reptile tins out, which again are being monitored between March and October. And this is the data for 2021, where we had 38 slow worms in the bison area, 4 viviparous lizards, and 1 grass snake, which is really nice to see. 57 slow worms in the domestic area, 2 viviparous lizards, and 30 slow worms, 58 common lizards, and 2 grass snakes in the control area. I've had a brief glance at this year's data, but it's not yet complete. I was told that there's still more data to come in. So I didn't want to include it because I didn't want to confuse anything by including data that isn't quite complete yet. But it looks very similar. If anything, Mike Phillips from Crag told me that it looks like there will be more records this year than there was last year, which again could be down to all sorts of reasons. But no matter the reason, it's nice to see a lot of records coming in. This is bioabundance. So as I've mentioned, we've got these SLAM traps out. That stands for sea, land, and air malaise trap between April and September. They're being they're being serviced once a month. So they've got these sample bottles attached to the top where the invertebrates fly into and are then being trapped. And then the volunteers go through those samples in the winter and sort them by order, so that we know how many flies, how many bugs, how many beetles for each month. And we have 5 traps in each treatment area, 1 in each habitat type. So looking at the different treatments, it's quite obvious to see that our control area has the highest number of invertebrates. In terms of habitat type, it usually is native region that comes out with the most invertebrates when you're just comparing the habitat types. In fact, in the control area, the native region came out with 2,633 invertebrates in July, which was a lot to go through for one sample pot. In terms of season or month, we had the highest numbers in July, and then it's sort of decreased in August and then decreased again in September, which is what you'd expect. Actually, this year, the traps have been out a little longer, so we're going to have more data. They've been put out in May, so we're going to have May, June, July, August, and September. And again, I would expect June and July to be highest in terms of numbers, and the rest of the months being slightly smaller in number. I can't include any data from this year yet because the lovely volunteers are still going through all of those samples, and it's probably going to take us until at least February to go through all of them. So again, this is something that will be included in the next monitoring talk, but there will be a report on this that goes up online if you're interested. I should probably also mention that the overall number we went through was 33,288 invertebrates, which is a lot. And I know some volunteers have probably lost the will to live looking at fly after fly after fly, but it is very much appreciated. And it is amazing to have this database and to look at all these records and numbers. Okay, category 3 includes heath fritillary surveys, that being counts, and also heath fritillary food plant monitoring. It includes wild herbivore surveys for us to establish what the existing grazing and browsing pressure is in the project area. It also includes the spatial mapping of which areas are being used and influenced by the bison and what their population dynamics look like. Now, this I won't talk about in this talk because we don't have the data yet, mainly because we don't have all the grazing animals on site yet, and for that reason they're not collared, so we don't have any data on how they move just yet. Again, obviously once the grazers are on site, they will be collared and we'll be able to look at that, and this is definitely something that is going to come up with a lot of really interesting and useful information, but will probably be in a good few months' time before we can even start to look at all of that. And then lastly, reducing human influence versus biodiversity uplift. This is where we're looking at how this nature-based solution is basically taking on the human's job of managing the woodland. So ideally over time, we're going to see that human input decrease whilst we see the biodiversity and bioabundance increase. This is something that we've only just started having initial chats about. It was very low on my priority list, mainly because it's not something that I needed to do before the animals arrive. I can obviously look at the data and get all those numbers retrospectively. So whilst I will get around to it, I haven't really started on it yet, so bear with me. But again, this is something that will be included in the future and will actually be really interesting to look at. It just wasn't very pressing until now. Heath fritillary. So as I've mentioned, butterfly conservation are doing the heath fritillary counts for us, whereas we do the food plant monitoring. So again, we have an army of volunteers out. Each volunteer gets allocated a survey area, and then in that survey area, they record the food plant abundance. So they're looking at common cowweed and ripwort plantain, which are both heath fritillary food plants. And this is a map that butterfly conservation got together for us. So you can see the dark points. Obviously, the bigger the point, the more heath fritillaries, and then the darker the yellow-orange color, the more common cowweed. And you can see that really nice correlation of where there's a lot of common cowweed, there is a lot of heath fritillaries. Although you can also see a few heath fritillaries down here where there actually isn't any common cowweed, or up here. So then it's quite nice to look at the ripwort plantain map, and you will find that actually that's where you find the other food plant. Again, this is the data for 2021. Butterfly conservation haven't gotten around to getting that together for us again. They had a very busy summer as well, so we're still waiting for that to come through. But just to have a quick look at this year's common cowweed mapping, there's a few areas that are missing, unfortunately. Again, for various reasons, mainly because, you know, volunteers just haven't had as much time as they would have hoped, or fell ill actually and just didn't manage to do what they thought they could do. And those things just happen. Also, I don't think it's a big worry because it's still part of the baseline, and we had a very extensive survey last year. So combining those 2 years is going to be a lot of data. That is definitely enough. And then obviously as of next year, we'll make sure that the entire area is covered again when it comes to monitoring heath fritillary food plants. This is the map of the deer transects. So again, you can see there's one going here in the control area, 3 in the bison, and 3 in the proxy area. And this little cross up here marks the only place where I found deer pellets. They were muntjac pellets. There's a few crosses down here. They were all rabbit droppings, so have been disregarded from this monitoring approach. And then having found that one pellet group, there are a few calculations that you can do. So taking into account muntjac defecation rates and mean decay rates, it means that we have a deer density of 1 per 100 hectares on site. Considering the project area is less than 600 hectares, we can assume that there's no more than 6 muntjac deer grazing across all 3 treatments with no other deer species present, which is kind of what we would have expected. It's not something that we see an awful lot, and it's something we already know isn't particularly common in the Blean. And that takes me to our 4th big banner, ecosystem services and natural capital, where we're looking at soil health function. soil health and function, sorry. And under that, we've done soil carbon surveys. So in all of my 145 vegetation monitoring points, I've taken soil samples, which have then been sent off to a lab to do soil carbon, to get a number for soil carbon. I haven't included that data yet. It's online if you wanted to have a look, but I thought it'd be rather boring to look at that spreadsheet, because unless you know an awful lot about soil carbon, you're just going to see a lot of numbers that probably won't mean much to you. Again, this is something that'll be really interesting to look at over the course of the next 5 years. So this is a survey that will be repeated 5 years from the baseline, so 4 years from now. And that will be super interesting to see how that changes and what impact the grazers have on soil carbon. And in terms of soil eDNA, so this is something that Canterbury Christchurch University are looking at for us. So the interns have taken all of the samples, they've extracted the DNA, and now the next step is for the DNA to get sequenced. So we just need to decide on what primers to use, which means we need to decide what exactly it is we want to look at when it comes to soil DNA. And what we definitely want to look at is fungus and bacteria, potentially also invertebrates, potentially nematodes. We'll have to have a look and figure out what the best solution is. But that hasn't been done yet, so there is no data to look at. Again, check back, it will be there at one point. In terms of pollinators, as I've mentioned, this is something that is going to be coming out of other surveys, in this case, very much the Standardised Invertebrate Survey. And Graham Lyons found 19 butterfly species, 3 of which had conservation status, which were the heath fritillary, small heath, and white admiral. Moths were very well represented when he did his survey with 114 species. 6 had conservation status. He found 63 species of fly, 2 with conservation status. Your Coleoptera were the most species rich, so that's the beetles, found most species in that order with 223 recorded, 22 with conservation status. And then when it comes to your bees, ants, and wasps, that's probably the order that was the least well represented. He found 21 species of bee, 11 ant species, and 2 wasp species, which kind of reflects how little forage there is in woodlands, with bees particularly being interested in open areas, which are very limited in the Blean. But again, something that hopefully the bison will help us create more of by all of the fantastic things they do. Speaking of which, there is just a little video of one of our younger females here who seems quite hungry and is going to do some bark stripping. So she's just finding a little bit that she can get her teeth under, and then she'll pull that bark off. And actually, I was just out on Monday with one of our lovely bison rangers, and I've seen the impact that they've made in the woodland already. They have debarked a lot of trees. They've taken a lot of trees down just by walking through the woods. And so I can really see how they will create a lot of this really lovely standing deadwood, which will be fantastic for invertebrates. I can see how they will create a lot of open space, which again is going to be fantastic for invertebrates. More sunlight coming onto the woodland floor, more native species being able to seed and disperse and grow. So again, it'll be really exciting to watch this over the next few years. This is the monitoring webpage, as I've mentioned. So if you're interested, make a note of this and have a little look. All of the reports that are finished, all of the data that we've got should be on there. And that is updated regularly. So if you're looking for something in particular that's not there yet, keep checking back. It should come ASAP. If you're missing anything specific or you have any questions around any of this, feel free to send us an email at blean@wilding.org.uk. And that leaves me to say nothing but thank you very much for coming and for being interested in all of the monitoring that's going on.